Okay. Okay. Hello, everyone. Welcome to the Data-Centric Biological Design and Engineering Seminar Series, which is part of the interest group activities of Alan Turing Institute. So this is the first one in our whole seminar series. And I'm Yuxin Shen from University of Edinburgh. Please allow me to introduce what we do and who we are on behalf of our organizing group before we have our amazing speaker today. Okay, so our DCB interest group aims to answer the key question: how can AI support the practical engineering of biology to tackle global challenges? And we are now organizing an online seminar series as part of the interest group activities. We want to cover 4 main topics: protein design and genotype-phenotype mapping, automation of biological systems, and physically informed biodesign, and finally, genomics and microbial engineering with big data. And we have our website here. Please check this. Please scan this QR code for our website. And it hosts all these information, including upcoming and past talk information, Zoom links, speaker schedules, contact details, and links to individual and series-wise calendar events. So do check our websites. And if you like this seminar series, please share with the people around you. So let me talk about our organizers. This seminar series is organized by Pedro and I and 3 PIs. So Professor Thomas Gartrowski, Professor Chris Barnes, and Dr. Diego Arzun. If any of you are here, you can just open the camera and say hi to the audience, maybe. Pedro?
Hi, yeah, I'm sorry. I'm Pedro.
And just any of you just say hi to the audience.
Hi everyone, I'm Chris at UCL. But Pedro and Yushin have organised this. They're the people who deserve the thanks.
Yeah, hi there. It's Tom from University of Bristol. Yeah, as Chris says, I think Pedro and Jizin have done an amazing job. So yeah, looking forward to the first talk.
Okay, so we already have a list of amazing speakers, and we are planning to have more. Again, this is all on our website, so don't forget to check it. And today, it is our great honor to have our first speaker, Dr. Stephen Goldrick from UCL. Dr. Goldrick is working on the digitalization of bioprocess and will tell us about next generation lead clone selection for cell line development through AI and machine learning. Welcome, Dr. Goldrick.
Yeah, thanks very much for the introduction, and thanks again for giving me the opportunity to present. So just to confirm, you can see my slides at the moment?
Yes, we can.
Excellent. Yeah, so then today's talk is going to focus on then digitalization of cell culture operations, enabling better clone selection and scale up during cell line development. So this work was basically part of a UCL AstraZeneca Center of Excellence. So this is a collaboration that we've set up between the Department of Biochemical Engineering and UCL and AstraZeneca. And the idea of the center is that basically we look at both process development, manufacturing operations from cell culture all the way down to purification. We also look at capacity planning and integrated process. And the idea is leveraging advanced scale down and modeling tools with the ultimate aim of generating basically integrated set of predictive tools for the whole bioprocess optimization and control. So the talk today I'm going to focus on is going to leverage some of the microscale high throughput studies, predictive correlations, and multivariate data analysis. So today's talk is going to focus on the challenge related to cell line development. So cell line development is a hugely important task when it comes to basically identifying the right clone for basically generating the right amount of therapeutics, and also the therapeutic basically has the right product quality for during manufacturing and scale up. So for those that aren't too familiar with cell line selection, it's actually quite a long and difficult task. So I'll basically briefly explain, if you know what I mean, the main steps involved in cell line development, and we'll talk about basically some of the limitations then of how the data is not fully utilized, and then basically we'll talk about then more improved ways that we can do it. So typically, basically, when it comes to screening clones, so normally basically what we do is that we screen anywhere between 1,000 to 10,000 clones. And this is basically then during the first scale of, during the first basically selection process, we're basically evaluating then all these clones on microplates or T-flasks. So we typically basically operate these clones in batch operation because we have quite a high number of clones. And due to the low volume in terms of the selection criteria that we'd select, basically the clones to move on to the next stage is mainly just based on productivity in terms of how well the cells grow. And then after we basically finished our kind of wide screen, we basically move on to suspension screens. This is basically where we kind of reduce the number of clones, and then we're down to a more manageable number between 100 and 1,000 clones. We typically basically operate this using shake well plates. So this is basically normally using about 30 mils of volume. So this can then also looking at then fed batch operations. So we can slightly extend the length of and evaluate the clones over a longer duration. And due to basically the larger volume then in terms of at this scale, we can look at both the productivity, so how well the cells are growing, how much product they're making, and also then basically, you know, looking at the quality of the product that is produced. So from this, then the next part is in terms of then we basically evaluate how the cells perform basically when we put them into a microbioreactor. So a microbioreactor is a scale down bioreactor system. Is very similar to large scale operation, and at this stage we can evaluate how the cells actually perform when they are when the pH is controlled, when they have you know, agitation. That's going to be similar to scale up, and this is basically pretty much a very good accurate mimic of what we'd expect at the large scale. So this is basically where, in terms of, in addition to the productivity, product quality, we can also basically collect some, you know, time series measurements, which offline measurements, which can be used basically, which can be used for in our decision-making process. But one of the things that you will notice basically during all of this basically stages, we don't necessarily often look at then the inline high frequency measurements, and that's something I'm going to come back to then later on. But once you've basically completed these screenings, what we typically have then is basically one clone, and you might basically have a backup clone just in case you basically do see any issues. And this clone effectively then ultimately should display high productivity, ensure all the product qualities are within desired range. So once we have the lead clone in terms of then, so yeah, this process can take anywhere between 20 to 25 weeks, because it's quite a long commitment, and obviously then takes a significant amount of time when it comes to then how much time is involved. So it's a significant investment of time. But once we have our lead clone, then the next stage is terms of evaluating then the lead clone. So what we typically do is that we would basically take the lead clone, and then we do an early generation bioreactor clone. So we basically evaluate the clone's performance at a larger scale than the high throughput bioreactor. So typically we do this in about 5-liter systems. And the idea of this is that then, in terms of, we can evaluate how the clone performs at a, you know, a better scale in comparison. So that the 5-liter system is more comparable, say, to a 20,000-liter bioreactor. And the next part of the clone evaluation is the stability study. So more often than not, we need to basically identify, when you're basically thinking of commercial scale manufacturing, the cells go through a large number of passages. So we want to make sure that the clone that we have selected in terms of can deliver stable and maintain that productivity when we do, you know, a couple more passages, which is typical then for commercial manufacturing. So in this case, basically, we would passage the cells a large number of times and check the stability during each stage to make sure then that the clone that we have selected is stable, basically, and then can basically and is fit for purpose at the larger scale. And the final basic part then, in terms of looking at then, is just doing a large scale and downstream DSP, which is downstream process validation. So obviously then previously we were only operating with, you know, 5 to 10 liters, and that is quite limited then in terms of the volume that we have to really assess our downstream processing. So what we typically do is we'd run 250 liters or maybe 200-liter fed batch operation, and this allows us then to generate significant material to evaluate then our downstream process operation, and also then this generates then enough material for toxicology. So again, this time frame can take anywhere between 7 to 10 weeks. So overall, you can see there's a lot of basically time and material requirements basically for this cell line development. So to summarize in terms of then, what we typically see then in terms of some of the challenges when it comes to cell line development is that we basically have a lot of data, but we actually don't really use much of the data which is generated. So obviously, as we increase in scale in terms of the number of candidates that we have data for, will obviously then reduce. And then in terms of based on the nature of like research and development, the green line basically shows in terms of the number of clones that we would basically have available at the different scales. So these basically peak in around like the microbioreactor scale or the small scale bioreactor runs. And then finally, in terms of what's important in terms of looking at then the data that is generated at each of these scales. So obviously at the very, very small scale, 1 mil scale, we don't have any of the online measurements, but once we get to the microbioreactor scale, we would typically have all of the online, offline, like the temperature, pH values. So that means then that we would basically have a significant amount of online and offline data recorded at these various scales. So if you basically multiply the data recorded by the actual number of cell culture runs, what we have is basically the available information. And then what you can kind of clearly see is that there's a bit of a sweet spot. So if you look basically in terms of where we have a large number of cell culture runs and all of the available online and offline information, we can see that we had the most amount of information recorded in the microbioreactor scale. And this is basically then what has been the focus then of this particular study. However, one of the things that I highlighted earlier in terms of when it comes to cell line development, that we would typically only basically use the productivity, product quality, growth, and offline data. So this information is kind of recorded typically every 24 hours or is basically only recorded at the end of the run. So if you summarize that information in terms of it basically accounts for 10 kilobytes of information. Whereas if you consider then the online data, so all of the pH, dissolved oxygen, gas flow rates, all this information that is recorded for each of the bioreactor runs, this accounts for about basically 10,000 kilobytes of information. So this data is often ignored, and so effectively we're only actually using 0.01% of available information, which is actually used for lead clone selection. So the research question that we focus on here, so can we make better use of this available data to select a lead clone and that's more suitable for manufacture and scale up? So the question is, why do we only utilize 0.1% of the available information? And the reason that we do that is because we're basically, very used to handling in terms of endpoint measurements, such as say final titre measurements. So we basically are quite familiar with dealing and handling with, bar graphs. So effectively, when we have say the endpoint titre measurements or maximum viable cell concentration or aggregation, we can do a bar graph. We can kind of quite easily basically then interpret it. So when we have these kind of endpoint measurements, they're easy to visualize, easy to interpret, easy to quantify, and then it's easy to evaluate the best cell line. However, when we look at the time series data in terms of this, actually becomes quite challenging. So the other 99.9% of the information is basically shown here. So this is a typical pH graph, but looking at this information, it's very difficult to basically understand what's a good pH profile, what's a bad pH profile, is it well controlled. So it's difficult to visualize, difficult to interpret, difficult to quantify. In fact, it's quite difficult to use in a decision-making process. So when we are basically then using then the univariate data in terms of, we more often than not in terms of would focus on then just doing minimum maximum targets for specific for specified attributes. So we basically typically just do a univariate approach. So in terms of what we do is want to maximize the titer, minimize aggregation, minimize lactate, and say maximize our productivity. So when it comes to basically selecting the best clone, we can just identify in terms of what clones perform the best when it comes to basically looking at different attributes. However, when we have a lot of basically different data being recorded, it becomes a multi-criteria decision making. That actually becomes quite difficult to understand in terms of a challenge to reconcile in terms of conflicting attributes for multiple different cell lines and formalize the decision on the lead clone. So one of the things then that we focus on was basically how do we leverage all of this data for better decision making. So what we basically developed then with AstraZeneca was this thing called Cell Line Development 4, so CLD4. So it's basically the next generation cell line development for Industry 4.0. So it's CLD4, for Industry 4.0, and has basically 4 steps. So the first step is data classification, feature engineering, and storage. The second one is then developing a cell line manufacturability index, which allows us then to speed up basically and quantify the optimal clone. The third step is on data analytics, and the final step is then on autonomous report generation. So I'll basically go through each of these steps, how they can work, and then basically we'll go through a case study then at the end. So the first step is just looking at then the data classification. So within the data being recorded, it's quite complex in terms of the different, all the data can be kind of broken down into, obviously it's recorded different frequencies, has different formats. And, you know, an example of then what we can do is we can break this data into 5 different classifications, which are related to the CLE4. So we can have the process parameters, which is the high frequency online data. This is typically recorded in multiple CSV files. We might have the growth data, which can be related to all variables related to the cellular growth. This is typically recorded in, say, CSV files. And then we might have the productivity, which would be offline. It might be endpoint, or it might be time series data. And then this is typically, again, recorded from different analytical devices. And then we have the product quality, which, again, you know, depending on how this data is recorded, it might be basically then, you know, stored in different Excel files. And the final kind of information, which is the meta information, is the non-numerical data recorded. So in terms of cell culture observations and, say, predicted cell markets. So if we can break down the data into these classifications, it becomes a little bit easier then to move forward with basically evaluating them at a later stage. So the question that I kind of mentioned earlier in terms of how do we summarize the time series data? So looking at the pH profiles that we saw earlier, in terms of these are quite difficult then to evaluate. So what we typically do then in terms of, and this is quite a useful way to break down this all these measurements. So the pH and temperature can have anywhere between 10,000 data points for each bioreactor. So it's quite a lot of information. But what we can do with this data set is actually, if we consider then some basic statistics that actually can accurately describe the data set. So we can basically then, in terms of, reduce the pH, temperature, or DO. So we can take the maximum, minimum data points, the endpoint, average pH, standard deviation, time above and below set points. So as an example though, so the pH recorded for an individual AMBER 15 system, which is a high throughput bioreactor, typically contains about 15,000 data points. This can be summarized into just 6 new features. So we take the maximum, minimum, endpoint, average, standard deviation, and time above and below set point. And the time above and below set point can be important depending to better understand basically process control. So this can basically result in a 2,500-fold reduction in the data. So that's kind of useful then for temperature, pH, and dissolved oxygen. When it comes to basically, say, the gas flow rates, so again, looking at the gas flow rates for dissolved oxygen is quite difficult to basically understand then, you know, how basically dissolved oxygen will impact the process. But what's a useful metric is if we take the cumulative sum of how much of the gas flow rates, we can basically then take the endpoint cumulative value. So this basically then demonstrates that this information will tell us how much oxygen these cells required, because you might basically select a clone which gave us the highest titer and highest viable cell concentration, but actually consumed twice as much oxygen as previous cell lines. And then that actually means when you scale up your process, you might be able to deliver that high amount of oxygen. So it's a very important design criteria, but it's very difficult to get that information looking at the gas flow rate. But if you take the cumulative sum, again, it allows us then to break that down into, you know, easier metrics, which we can use then in our data analysis. So the final kind of part then of the step 1 is in the storage. So one of the things that we typically do is obviously we talk about data lakes. So this is where we have the large data, the unstructured raw formats, so multiple different CSVs. So this actually becomes quite difficult to analyze. But what we can do, and again, is we can break that down to a data warehouse. So this basically large data, then it's a kind of structured, more processed data set, but still it's actually quite difficult to handle this data set, you know, on a home laptop. But what we can do is we can take the key statistics, important features, and the summary analysis, and what we have then, you know, use that, what we call is basically the data lakehouse. So effectively it's just, you know, breaking down the data into more digestible bits of information, and then once we have it in that, then that we can basically easily analyze without the need for high computational power. And it also then, you know, reflects in terms of then, you know, all of the raw information that was shown in either the data warehouse or in the data lake. So the next step then in terms of is to help with this complicated decision making, we developed a new metric referred to as the cell line manufacturability index. So this can greatly simplify the ranking of the cell lines and handle anywhere between 10 or 100 different attributes, provided they're all available. So the metric basically is based on a weighted sum method and requires a weighting of importance for each attribute and a decisional and a dimensionless ranking for each attribute, say I for each cell line J. And effectively what we can do in terms of is that, so to kind of give an example of what this looks like. So if we basically had 12 different cell lines, then in terms of, we can apply basically a weighting in terms of which variable is more important. So that's basically a multiplication figure. But what we'd want to do is that we can calculate the dimensionless ranking of each attribute for each of the cell lines. So looking at then, for example, if I just get my laser pointer. So if we want to basically then maximize our titre concentration, then the cell line number 8 basically has the highest dimensionless ranking compared to all the other cell lines. That's basically a value of 0.17. So it's 0.17 because we basically have 6 different variables, and then that's basically 6 divided by 1. So the maximum value that can have is going to be 0.17. But with this dimensionless ranking, we can break down the raw information into this dimensionless rating, multiply it then by our weightings, and then in terms of, we can then have our cell line manufacturability index, which allows us then to get a single metric which can quantify all this information. So the only thing we need to decide in terms of the weighting, so how important each variable is, and then also in terms of then whether, we want to maximize and minimize the variable. So obviously for titre, for example, we want to maximize the titre, but for aggregation, we'd want to minimize that. Hence, in terms of looking at the ranking, say for the aggregation, all the variables that had a value of 0 all had a dimensionless rating of 0.17, which is the maximum that can be achieved for each of these particular variables. So defining the weighting of each attribute is of primary importance. So this can be highly dependent on the mode of action molecule. So what we basically this obviously where we work quite closely with our scientists at AstraZeneca. So we can have adjust the weightings that can be either project, cell line, or molecule specific. So sometimes, depending on if it's mAb, or a fragment antibody, or bispecific, we might basically have different requirements. So a quick example of looking at then, if we calculated the manufacturability index for 40 different cell lines producing a novel antibody peptide fusion, which is shown below. If we have equal rating, then in terms of where we have our product quality is as important as our productivity and our growth, we would select in this case cell line 8. Whereas if we are basically more worried about basically product quality and want to maximize, then ensures that we want to select a cell line that's going to be easier to purify. We aren't maybe as interested in the cell growth, then that means we basically, if we add additional weights to product quality and productivity, we can actually then adjust the weights, and that will give us that would basically recommend like cell line 48. So ultimately, the cell line manufacturability index quantifies all the information into a single metric, and it allows us for easy evaluation of multiple cell lines and the corresponding attributes. So the next stage in terms of I mentioned about the benefit of breaking down the raw data into individuals, and important metrics. And this is where we can basically do some data analysis. So the principal component, for example, then can be used to look at then correlation. We can easily visualize then the productivity growth, product quality. They can be easily visualized, and then also we can identify if there's any cause and effect relationships, and then that can be subsequently investigated. So in this example here, we just have the 48 different clones. We can see these top 5 clones and all basically, grouped together, and that's obviously then related then to the titer and the viable cell. Both these clones all had high titer and high viable cell concentration. But by having the productivity growth, product quality, and process parameters, we can relate them in terms of if there was any particular issues with the bioreactor, like the pH growth or the temperature, and then if that had an influence on the product quality, then that will obviously then raise some, these correlations can be identified using the data analysis procedure. So the final part then basically then in terms of obviously a lot of information here, trying to basically digest all of this can be a little bit tricky. So there's obviously been a lot of interest then with, human readable reports. So obviously then, ChatGPT at the moment in terms of has been used quite consistently. So this is a stochastic natural language generation. So again, this is kind of great for say cheating on your homework, but is inconsistent when it comes to reporting on bioprocess scientists, because when we generate a batch record, we want to basically have a consistent template. So what we basically did was generated a automated a natural language generation rule-based template that was able to basically generate a cell line development report. So the document text is kind of shown here is highlighted in black. So that's basically then fixed text. So this is basically a rule-based template. And then basically we can have these user inputs. So in this case, we can basically adjust the template. So we want to basically have a template for, say, 5 different clones. Then this will basically say in terms of, okay, the top 5 clones were, and then basically this will pull the information from our data lake, which will basically be the top 5 clones based on manufacturability index were 8, 19, 3, 10, and 4. The lead clone, which is 10, which is obviously going to be the one that had the highest cell line manufacturability index, then had a max titer of 5.2. And this is basically where we have that rule-based template. We can kind of write basically automatic scripts that can basically pull that information, and then that will allow us then to effectively digest all the information from our data lake into a human-readable report, which can be very can be very useful to share with scientists or even upper management that can simplify in terms of that why we selected this clone, and then in terms of kind of move more towards data-driven selection when it comes to the lead clone selection. So obviously this sounds like a kind of lot of extra work. So, in terms of working with AZ, they want things simple. So the question is like, do we really need all this additional analysis? So in one of the case studies that we looked at then, so there was a novel antibody peptide fusion. So this was basically carried out within AstraZeneca. So during R&D activities, there was led to discovery of a product-related variant on a novel antibody peptide fusion. So during the size exclusion chromatography, we could see this basically shoulder on one of the antibodies that was being generated. And this basically shoulder was identified as effectively then looking as being trisulfide bond. And this actually then had a significant impact on how the actual antibody then performed and affect the efficacy quite significantly. So in terms of, to mitigate this risk then in terms of what the company did was that they basically then, okay, so we just said, let's just try to generate an additional parental clone screening was carried out to ensure that the selected cell line was not a producer of trisulfide bonds. So what we basically did was basically trying to basically then select a clone that didn't basically produce trisulfide bonds. So during the initial cell line development, this was basically a summary of all the trisulfide bonds. So it's a bit difficult to see, but effectively what you can see is that there's a number of different clones that didn't actually produce any trisulfide bond. And when these were basically subsequently re-cloned again, then in terms of we could see then now in another microbiota screen that we were able to basically identify a single clone that had 0 trisulfide bonds. So the company then were quite happy with saying, okay, we basically were able to basically modify our cloning procedure, and then we have now selected the clone that doesn't basically have any trisulfide bond based on these two screening. However, basically when they evaluated a clone at the 5-liter scale, there was no trisulfide bond. 50-liter scale, no trisulfide bond. It was only when they got to the 250-liter scale that basically then they had basically trisulfide bond issues. And this basically then in terms of effectively then means that the trisulfide bond was identified at scale up. And the question is then, could the CLD4 be used to identify this issue a little bit earlier in the process? So what we did was then we took all the available information. So again, kind of using the first step was basically getting all of the titre, VCD, all of the online, offline, so the productivity growth, product quality, endpoint, and process information. We then done our feature engineering, so calculate a base year maximum, endpoint, all of the things I mentioned earlier. And then with that, then in terms of that was just kind of summarizing all the information. Then we have to basically then, apply this then to our cell line manufacturability index. So in this case, we're just showing the top basically 12 and bottom 12 clones based on a dimensionless ranking of the data from the 20 different variables. So we have all of these variables that we're interested in, and then we have, in this case, we increased our weight of trisulfide bond to try to basically ensure that we select a clone that will basically then minimize, that one ensures that we basically the clone will not have any trisulfide bond present. So this basically then, allowed us then to give us a quite a, able to basically select then in terms of what were the best clones based on all these different variables considered. And then we're just looking at then, when we looked at then just looking at the raw data, you can kind of clearly see that obviously just the cell line manufacturability index selected these clones based on the highest manufacturability index, and then looking at the raw data, it's not always basically clear in terms of why in terms of then that these are selected. So for example, previously they might select some of these other clones, which is shown here, they might have higher titre, but if you consider all of the titre, lactate, all the online offline data, then you can actually identify better clones that will basically perform better when it comes to scale up. So the top clones then in this case were 8, 19, 3, 10, and 4. And then again, when we done our data analysis, this is where it actually became a bit interesting, because we could clearly see in terms of then that there was actually a clear clustering between those cell lines that had high trisulfide bond and those cell lines that had low trisulfide bond. And one of the things then that we were able to look at was that the trisulfide bond here is shown here by the as a product quality, and you can see that there's quite basically strong correlation between trisulfide bond and the pH. So the pH then statistics that we calculated with the pH basically time above and below set point, as well as the pH mean, and then the time between, say, the average between day 0 and 7. So this basically then means that potentially the trisulfide bond wasn't actually a clone issue. This was more of a process issue, and this was something that this was able to basically clearly see that we were able to pull this out. And then if we just kind of delve into that a little bit deeper, we can obviously then generate the correlation table, which kind of summarizes the information from the PCA in a very similar way, but actually quantifies in terms of how strong that correlation was. So by analyzing the correlation table, we can see that the trisulfide bond here then in terms of really has quite strong correlation between then the mean pH of the process, as well as basically then pH then time above and below the set point. So once you then delve into data a little bit more detail, when I kind of showed you earlier, when you just basically look at the raw pH data, it's actually quite difficult to kind of see any of those trends. But even when you plot, say, the pH mean versus trisulfide bond, you can kind of see that, okay, maybe there's a little bit kind of then the higher pH means does result in higher trisulfide bond, but it's only kind of when you really kind of color code basically those cell lines that had high trisulfide bond compared to those that had low trisulfide bond, and you can clearly see in terms of that the pH profile significantly influenced basically the trisulfide bond. So therefore the need for improved pH control was required, and this was actually then quite an important then piece of work, because it basically demonstrated then that we really need to basically consider basically the influence then of the pH when it comes to the clone selection. And we published this kind of work in the paper, showing some full details are there. So ultimately then in terms of that, the cell line development, if we apply that then to the initial parental screen where they saw the issue, we could have basically see then that we could have significantly basically then identified earlier on in a process development cycle, and this could have basically reduced the time and labor cost by 13 weeks. The resources that could have been avoided is 1 Amber 15 system, 18 5-liter bioreactors, 1 50-liter bioreactor, and 2 to 50-liter bioreactors. So that's obviously a lot of cost, a lot of money, and a lot of material. So ultimately then the cell line development approach can result in a significant amount of savings. So effectively then, the kind of coming to the end here. So we have the cell line development for then can leverage then, by building a data lake to store all the information, productivity, product quality, growth. This can then feed into then the cell line manufacturability index data analysis. And then with basically then the meta information, such as say target market, patient dose, we can basically then, use a natural language generation to generate an autonomous report. So one of the things I kind of didn't mention too much, but obviously once we have all of this, and once we have that data lake built, we can write algorithms that can automatically generate these reports, which can actually save a significant amount of time. And then can kind of, so in this case, so this report was able to basically analyze all that data automatically, but also then in terms of can also then highlight. So as an example here, we kind of said that the multivariate data analysis highlighted some concerns and product quality issues. So the average pH between day 0 and 7 correlation or squared of 0.8 with trisulfide bond, which is defined as a critical quality attribute and therefore requires further investigation. So basically, in addition to doing all the analysis, then we can automate basically the insights into the data which are collected, and that can be very, very valuable then to the scientists. Basically, after they finish their run, they can basically not only have all the data analyzed, they can also basically see some of the important insights once you set up this automatically this autonomous report, and then input basically some of the what your typical ranges are. In addition to that, then in terms of more often than not, when it comes to cell line development, people basically focus on just maximizing titer, but what companies are starting to realize in terms of is that, and this is basically quite important when it comes to different types of drugs. So once you actually know what drug is going to be developed, and then in terms of, and have an estimate of your patient base, we know what the titer is. We basically know what the annual dose requirement for the different patients is, so we can work out ultimately basically how much product is going to be produced per year. And this can be quite important then in terms of, by making some quite crude calculations about what is the typical required, say, capacity that we need to generate and deliver this material, that it can basically meet then demands of this particular, say, antibody or therapeutic protein. Like a good example of this is obviously then, say, Eli Lilly's basically new wonder drug. So in terms of, I think they struggle quite significantly then with the capacity planning, because I think their patient base, they had planned to basically, it wasn't planned for the current target market. So when basically then there was some changes, then the patient base increased significantly, and then they didn't actually have the capacity then to support the manufacture of this. But if you can consider that then in your cell line development, then obviously then, you can try to mitigate then some of these issues at the large scale. So in summary, then I think like the cell line development can basically hopefully enhance your process understanding, enable then for the best lead clone to be selected that will scale up more appropriately. So most information then is kind of summarized in the paper, which is shown here. So with that then, I'd just like to thank then all of the input then from the collaborators at AstraZeneca. So Matthew Cheeks, Richard Turner, Claire Lovelady, James Savory, Diane Hatton and Nick Bond, and then also my collaborators then at UCL, so Susie and Haneen. So and thanks again for everyone to dialing into the meeting today, and happy to take on any questions.
Well, thank you very much for that talk. It was really interesting. I see we already have some questions. Thomas Gorojowski is asking, based on these vast data sets, do you see counterintuitive results on conflicting outcomes that might require new experiments? If so, is that something that could be used to design useful experiments to better understand these production processes?
I think it's something which I think definitely AI is could be useful to better design the processes. So I think at the moment, when it comes to, say, cell line development, they have like almost standard operating procedures. So they are collecting the routine data. But I think then what even this work showed was that if you actually make better use of the data recorded, you can do probably smaller amounts of experiments. So you could design the experiments better to ensure that you will select a better clone that will scale up more appropriately. And that's something that, AI can definitely support with. So you can, that in the long term, if you know what I mean, if you have more information, especially with some of these new sensors, then that means you could generate cleverer, smaller design of experiments, and then get better clones, but hopefully then less experimental work. So hopefully that answers your question.
Do you have a question?
Hi, thanks, Stephen. I have a question. It's cool talk. So I guess two questions. First is like a very naive one. What's the difference between the cell lines? Is it genetic or just phenotypic? Or
so it's just genetic, if you know what I mean. Like, so in terms of the random mutations, and that's it's thinking about it, obviously they just try to screen a large number of different clones and hope to get a good one. But in terms of, there's a lot of then synthetic biology which really could significantly enhance some of this, rather than just screening a large number and hopefully that you'll get a high producer. But that's what they do. It's a numbers game. But there's definitely massive room for improvement here, I think.
And then my second question is, is it possible to get like some outcome information? So it seems like you're designing the selection procedure without knowing exactly what without having whether a clone is successful or not at the end. I mean, you had one case study where you could definitely change those weights. So, you know, is there a way to, if with more data in the future, would there be a way to optimize those weights, for example, rather than doing it by?
I think that's kind of why you kind of need the input from the scientists. So in terms of, they kind of know in terms of, you know, what are the main issues they have at scale up. So, you know, they might get high lactate, which can, you know, have a big impact then at the larger scale. So they could kind of minimize for that. So that's kind of where in terms of then you need that operator input. But if you had a large amount of data, then effectively you could automate how you design those weights more appropriately.
Okay, yeah, thanks.
I do have a question, Stephen. So right now the index is a weighted sum, right? So are you planning eventually to do this like a non-linear index in case you see some interactions or some non-dependency between the different criteria?
Yeah, so I think one of the main criteria with this particular work was ensuring then that you have transparency on the clone that was selected. So in terms of, we didn't want to have a black box approach. We kind of saying, here's your inputs in terms of, here's your weight. So in terms of the scientists that actually then typically would select a cell line, almost they kind of apply this metric, but not necessarily as consistently. They'll go out, this had the highest titer, this had low lactate. So they'll basically select it kind of almost in a manual way. This way kind of just gives it more of a data-driven approach. So, you know, it can be a little bit more useful. I think you are kind of right that there are kind of then definitely some, you know, more complex ways that it could be doing, but then you kind of lose the interpretability of the metric. So it isn't as valuable then when it comes to, say, some of the scientists that are doing the experiments, because they want to, you know, fully understand basically why this was chosen and not necessarily because the algorithm said so.
Thank you.
Yeah, there are also questions in the chat box. I think from Yi Hon Cho, there's a question to follow up on Chris' question: is there genomic data for each cell line that can be incorporated into the data analysis? Do you think having those data would be useful?
Yeah, so there wasn't that data available, but I do agree in terms of that information would be really, really valuable, and that could be something then that could, you know, really enhance in terms of the way that these cell lines are selected.
Yeah.
Thank you.
Yeah, and another question is there a limit on parameters would you need to build a metric for example, a better cell line?
Yeah, so I think in this case we just used 20 different parameters, but that was kind of the features I created, based on, you know, looking at, say, the pH profiles, what was considered important, like the mean, standard deviation, so whether it's above or below a set point, you know. But you could collect a lot more metrics that are available, a lot more data that could be included. So you could basically add additional information to this, and then that could result in better cell lines. But again, it's kind of back to the point I made earlier, trying to keep things a little bit more interpretable. So it worked well when we had 20 variables, but you could then expand that. You know, so mentioned earlier, if you had some genomic data, then, you know, that could also be included. So the metric itself kind of, you know, it can handle, can handle multiple more metrics. So you could make it more and more complex, but then you kind of lose some of the interpretability.
Thank you.
Thank you.
I do have another question, Stephen. What was your biggest hurdle while trying to manage all these different data types and data information? I imagine it must be very complex trying to do all of this. And if you're planning on using any data standards, or if you are using any data standards to store these.
Yeah, I think that's kind of quite a good point. I think it's something which, you know, even like the pharmaceutical sector as an industry is not necessarily the best when it comes to data standards, because a lot of times, you know, the information was recorded on an Excel sheet that was then modified and then sent, you know, for analysis. So I kind of had some automated scripts which I could analyze, say, the raw information coming from, say, the high throughput bioreactors, which is kind of always in a standardized format. So that was a way to, easily calculate information in an automatic way. But when you're dealing with Excel sheets that are modified by individuals, it's very, very difficult then to, standardize the way that your algorithms will extract information. So I think it's something which, we struggle actually quite a bit with, and we're kind of saying, once you define how you're going to, save this data, that is consistently saved that way, because that will simplify then when it comes to the automation of this procedure. But more often than not, yeah, we had to always kind of slightly go back to the Excel file and change around certain things to make it kind of fit to what was done previously. So I think it's something which, as everyone kind of says, in terms of when it comes to data science, 80% is kind of data wrangling, 20% is model building. And I think that was basically pretty much a core part of this. I spent a lot of time getting the data, bringing it together, doing some wrangling of it to get it in a format that was suitable then for the automation. So that was kind of, yeah, I think it's something which the data standardization could significantly enhance.
Thank you. Any other questions? I see a raised hand. Oh, Chris.
Sorry, I had one last question, and what do you think they would be interested in making? I mean, this is probably a no, but do you think that this sounds like it could be, you could make a competition out of some data like this, and let people try and develop new approaches? Do you think they would be keen or willing to make their data available?
So some of the information I think could be made available, but I think some of the data is proprietary, so it is that little bit kind of challenging. But I think one of the other kind of issues is that we don't actually kind of fully know the answer to what is the best cell line, because we only basically selected a certain number of ones that were scaled up. So even if we did then go back and say, actually, we should have selected this one, we don't have the information to back that up. So we have done additional studies where we said, actually, if you went back and done this one, and we did demonstrate that does actually, does perform better than that what was selected, but that data more often than not isn't available. So it's kind of hard then to kind of demonstrate that, to prove that this actually is better.
So I guess they would need to implement it and then also keep track, right, of where it was successful and where it wasn't. I mean, will they implement it then? Have you convinced them to implement it?
I think it's, that's some the other thing, which is I think we've kind of got most things implemented up as far as the automated generated the report, because that's just a little bit, a bit too complicated trying to get into their, SOPs. But the data analysis and then the cell line selection, the cell line manufacturability index is now kind of part of their selection criteria, which I think is quite good. But then, hoping, kind of still working with them, that we can do that automated report, but it's just a little bit more practically hard to get that implemented.
Okay, thanks.
Any other questions? Well, thank you so much, Stephen, Dr. Goldrick, to come and talk and give this fantastic talk. It was really enlightening, and I thank everybody also for coming and uploading with your questions.
Yeah, and thanks again for the opportunity to present.
Please.
Yeah, thank you, Dr. Goldrick, and thank you, everyone.
Cheers.
Yeah. Please stay tuned for other talks that are coming for the next month. So just check out on the website, and you'll see all the information is going to be posted there, as always, with the best, the most updated information. Yeah, and do subscribe to the Google Calendar or this Outlook calendar, whatever you're using, and you can have a track of what's going on in all these seminar series. Yeah. Thank you again, Dr. Goldrick. Thank you.
Thanks, everyone. See ya.
Thank you. See you.